Ladies and gentlemen, friends and colleagues, I pose for you this afternoon a simple challenge, a quiz you might say. What's the difference between me and Angelina Jolie? Hard to tell, I'd say. We're really quite similar. Perhaps you might agree that my husband is better looking than hers. But you probably know, many of you, in this context, the difference between Angelina Jolie and myself that I'm bringing up has to do with our chance of developing breast cancer. Due to genetics, she had a very high chance of developing cancer and made the difficult decision to have a prophylactic mastectomy and ovarectomy. But what's the difference? What's the difference between us that gives her that chance of getting cancer and me, or anyone in that situation? To understand that, we have to look a little closer. Now, this picture also, you couldn't tell whether this is me doing a precision experiment or someone else doing personal grooming. But if we look at the cells, and in this artist's impression of a cell, we also look identical. But the difference between us is in one gene, in one piece of DNA, as was pointed out in the last presentation, at the nanoscale. A change in either the BRCA1 or BRCA2 genes causes a person to have a very high increased risk of developing cancer. Why is that? What does this BRCA2 protein, the part of that gene, do that prevents you from normally getting cancer? To understand that, we have to look at some real cells. So these are cells that have been treated so that they have broken chromosomes. And the pink spots mark the places where the chromosomes have been broken. In a normal cell, the chromosome breaks are repaired by a whole team of proteins. Those are the green spots, the proteins repairing those breaks in those cells. In a cell in which BRCA2 is not working, those proteins that repair the broken chromosomes never get to where they have to do their job. That's shown on the other side there, no green spots. Now, breaks happen in chromosomes all the time. They're normally repaired. If they're not repaired, the genome becomes unstable and sets a cell off on a path of potential cancerous transformation. So if the BRCA2 protein is responsible for bringing a team of players to correct this problem and repair breaks, we have to understand how it moves about. So we set out to watch BRCA2 moving in cells. And to me, this movie is more exciting than Mr. and Mrs. Smith. You can't tell the difference, can you? This is an amazing movie. The little white spots you see bouncing around, this is a single cell nucleus, are individual BRCA2 proteins moving about in a cell. We can actually make movies of individual proteins moving in living cells. We can measure how fast they move, how far they move, how often they stop, and how this changes when they have to do their job. We then know what's different when the protein doesn't do its job properly. Now, to do this amazing experiment required a wide variety of expertise: molecular biology, cell biology, advanced instrumentation, microscopy, image analysis, and mathematical modeling. This is a tall order and combines disciplines often taught separately: biology, physics, mathematics. Now, can we combine these to provide an education for the scientists of today and tomorrow? And indeed, under the auspices of Medical Delta, the Erasmus University Medical Center, and the Technical University in Delft created the unique nanobiology program. And we describe this program, a bachelor and a master's program, as using the language of math and the concepts of physics to understand the complexity of biology. This is the information we need to understand health and disease and to make a difference in the future of healthcare. This entirely new program was started from scratch. I was involved in initiating this program some years ago and have been the director since it started. I coordinate a lot of enthusiastic people, and that's one thing that makes this a great success. And that's not a mistake. They were chosen that way. There's a whole team of teachers who develop courses, teach, and continually revise those courses to make our program better. The administrators at both institutes love the energy of this new endeavor, and we have a team of folks who do the scheduling, organization, and coordinate activities between our very different institutes. It's not an easy job, but it works because we want it to. because this is the right educational program to train scientists of the future to make a difference in biomedicine. It's been a huge success, not least because of all of the students who've entered our program. 73 in 2012, 79 in 2013, 91 in 2014, and 102 in 2015, which resulted in the world's first nanobiologists. Last October, we awarded 16 bachelor's degrees. These students and those who are following them are the people who are going to make a difference in healthcare in the future, and also in many other fields requiring the expertise they have. They have an inquisitive spirit, and they have the quantitative skills to understand complex problems and make sense of them and propose solutions. So you may ask, what's in this for Angelina Jolie? Enough about me in this program. What are the things I've told you about? What are they going to do and make a difference in how we treat cancer patients? Now, I can tell you that some of the fundamental information that has been gathered in our research group on the BRCA2 protein is being applied to improve cancer treatment today. But to be honest, when I think about what knowing the diffusion constant of BRCA2 will mean in medicine, I don't know. And it's important that I don't know. Fundamental scientists generate information, and we don't necessarily know how it will be used in the future. This picture has nothing to do with breast cancer. This is actually data describing the behavior of fundamental particles from the Large Hadron Collider. This is a representation of that information, and I don't understand this data. But this picture reminds me of some of the images from my own field of work. I think you may have seen a few of them in the previous slides. The kind of diagrams that are drawn to show the intricate interactions between genetic data or metabolomic data or population data come out in the same sort of diagrams. So maybe our students in the future will be able to process even more complex information to understand not the stuff of biology, but what it does, how it changes in concentration, position, structure, interactions, and make sense of that in completely new ways. I don't know exactly where they'll end up, but they'll make a difference in the field of biomedicine and many other places in science and fundamental work. So to me, the beauty of fundamental science is that we don't always know how it will be applied, but we do it anyways. Our students will succeed if they create new knowledge, and our society will flourish if we use that knowledge wisely. So I'm sure that our students will make a difference in the world in ways that we can't imagine today. Who knows? Maybe the film industry will be delighted not to have to hire a body double. But more importantly, maybe our students will be inspired by biology to create new energy transformers, to make machines that can process information the way our brains do, or was alluded to in the last talk, to take a simple collection of molecules and make it do something as intricate and beautiful as cell division. So I've introduced to you how we add education to the medical Delta palette with the example of nanobiology. I look forward in the future to this program being an inspiration for others. We don't have to follow in the footsteps of great institutes elsewhere. We lead by combining technology, medicine, fundamental science, and education to face successfully the challenges of the future. Ik heb gezegd.